Research and Innovation by Professor Sanjay Sane. As the belief goes, research and innovation is the backbone of any civilization and is imperial part of upgradation of the society. So, welcome you all for the today's session to ignite the young minds for this research and innovation. And now I welcome our principal, Dr. Vikas Subeddi, to say few words.
Good evening to one and all. On behalf of Royal Higher Education Society's Royal College of Arts, Science and Commerce, I welcome Honorable Speaker, Professor Dr. Sanjay Sane, Dean of Faculty, TIFR, Bangalore, Honorable Founder, Sir Professor A. Lakdawala, Vice Principal and Management Trustee, Professor Malika Butwala, Management Trustee Zainab Madam, Akila Madam, Afroz Madam, Dean Kamala Madam, my colleagues, and all the participants for the webinar on research and innovation. Research is always an exciting journey, a walk through the unknowns, through the ups and downs, new findings and learnings. It's not only the destination, but the journey is really exciting. In the research laboratory, mistakes are expected, respected, inspected, and corrected. Today's world is solely based on platform driven by innovation and out-of-box thinking. Brain is powerful when you use to its fullest capacities. I'm sure that this webinar will help you to stimulate and achieve these capabilities. I encourage you all to pursue the learning from this webinar and nurture the learnings to the fullest of your abilities. Wish you all the best for the future. Thank you.
Now I would request Dr. Chitra to introduce our guest of today, Professor Sanjay Sane.
Good evening to all. It gives me a lot of pleasure today to introduce Dr. Sanjay Sane to the audience, our current students. Let me begin by saying that Dr. Sane is currently professor at NCBS Bangalore, a part of Tata Institute of Fundamental Research. He is an eminent scientist in his field. And to speak about his background a little bit, he holds a bachelor's degree in physics from Sane Stephens College, University of Delhi, a master's degree in physics from University of Pune. From the University of Pune, he went on to University of California for his doctoral studies in the lab of Professor Michael Dickinson, where he studied aerodynamics of insect flight. He further completed his postdoctoral work at University of Washington, investigating the role of antennal mechanosensors in control and stability of insect flight. His areas of research include physics, the physics, neurobiology, and the ecophysiology of insect flight. The number of publications are too many. I have not put a number to it. The citations are above 7,000 plus. I would like to mention over here that he is among the world's top 2% most influential scientists. He is in the list, in this particular list, the world's top 2% most influential scientist that has been published by the Stanford University in the year 2020. I feel very proud to say that because Sanjay happens to be my batchmate. We passed out many years ago from the same school. We completed our matriculation in the same school many years ago. So I really feel proud about it when I say that he's among the top 2% most influential scientists as per the list published by Stanford University. Welcome, Sanjay, to this webinar on research and innovation. And thank you for accepting our invitation. And over to you.
Thank you, Chitra. Thank you, Dr. Vaidya. Thank you all. Let me see if I can share my screen. I think I need to be allowed to share my screen. So screen sharing is disabled at the moment. So the
I
host
think,
can-
Hussein,
yeah,
you
now
make
I can.
this. Okay.
No, I'm good. Yeah. Okay. And can you see my screen?
Yes.
Yes, we can see.
Okay.
Yes, sir. Yes, sir.
Thank you. Thank you. You know, very kind words. I thought I will talk today about, so you know, my sort of target audience here is students who are probably in their third year and who are thinking about research and its general role in life and in society. And I wanted to kind of speak to them particularly about you know, what research is and why it is so much fun to be part of. But before I did that, I thought what I'll do is I'll kind of introduce you to where I come from, my institute and my campus, because this is an integral part of you know, where research happens, and also I'll tell you a little bit about how these two are connected within a campus. So I live in Bangalore, and this is the aerial view of the broad campus, which is called the BLISC campus, the Bangalore Life Science Cluster campus. And this campus really consists of three institutions. One is us, the National Center for Biological Sciences. So you can see that there's a lot of green space, which I'm particularly happy about. But the labs are here. My lab is somewhere there. And these are two buildings. There's a swimming pool, hostel, etc. This institution is part of the same campus as another institution called Instem, which is the Institute of Stem Cell Research. And then there's a third institution called the Center for Cellular and Molecular Platforms. And I'll explain to you what these three institutions do and how their work is separate from each other, but also similar to each other. So my institution, the National Center for Biological Sciences, is part of the Tata Institute of Fundamental Research, as Chitra just mentioned, and we are under the Department of Atomic Energy, and our mandate is fundamental research. And I'll sort of explain why fundamental research. So when we talk about fundamental research, we are really looking at mechanisms of various things. And this is typically biological things, but they range from molecules all the way to ecosystems. And so we have scientists on campus that work in all of these different areas and sometimes in multiple areas. And there's also right here, if you can see my cursor, a building which is devoted to theoretical biology. And this building houses, I think, mostly physicists who are looking at biology from a sort of formal mathematical standpoint. And they are really doing really interesting things in that realm. So there's that. And then we are sort of a center for biology within the TIFR system. TIFR Colaba campus also has a biology center, but typically what has happened is that in Colaba, when space gets too constrained and groups grow beyond a critical mass, they go off and form their own campuses. So there's a radio astronomy campus in Pune, and there's NCBS in Bangalore. And then there's now a TIFR Hyderabad in Hyderabad. And so there are an HBCSC, of course, in Bombay, which is whose mandate is science communication. We have, I should emphasize that our culture is very multidisciplinary, meaning that you'll find a large number of faculty at the National Center for Biological Sciences who got their training either in chemistry or in physics or mathematics or other branches of science. And we're proud of this because we've sort of tried as much as possible to dismantle the sort of labels that we typically attached to science, and they're completely artificial. The Institute of Stem Cell Research and Regenerative Medicine, also called the Instem, is in the same campus. So this is located roughly, so somewhere there is NCBS, and then and it's from Instem that the last picture was taken. The Instem is relatively more recent, and it is under the Department of Biotechnology, but it's part of the same campus. So we work very closely together, but the mandate of Instem is translational research, meaning they're not, what we call, they're not doing fundamental science. They're doing science with the view of applying it to medicine and so on. However, one shouldn't really take these mandates too seriously either. I mean, there's enough scientists in NCBS who do translational work and several in Instem who also do basic science. It's really a very porous boundary between the two. Now, in modern times, the science is no more contained within your lab. In fact, so much of science is done outside of your lab. Be it if you want to do imaging, or if you want to do electron microscopy, or if you want to do any kind of mass spectrometry, things like that, you need facilities. And one of the major challenges that we had when this campus was put together was to put together facilities that were world-class facilities, which would be managed by top people who would then keep them up to date. And really, if you want to do science at the cutting edge, then what you need is really good facilities. And so one of our aims has been to disregard what our affiliations are, meaning the Department of Biotechnology or Department of Atomic Energy. So they have an MOU together, and what we do is that both institutions contribute to generating these facilities, which then form the nucleus in which science is done. So scientists from both institutions collaborate with each other, and their collaborations typically centered around such facilities. So who manages the facilities, and how can we get top people to come and manage the facilities at governmental salaries? Well, it turns out that's not needed, because the third institution, CCAMP, is an institution that was initiated by the Government of India, Department of Biotechnology, but it's a private entity. And so its mandate is to manage the facilities, or help us manage the facilities, but it's in suspension between Instem and NCBS. And because it's a private entity, they can offer very, very competitive salaries to the ones in the industry. And this also doubles up as a technology accelerator, meaning that it is where new companies can incubate. So if there is people with really good ideas for a product or a company, then they can approach CCAMP, and CCAMP will help them form these companies. And so this is the view of, I would say, a modern campus. And you'll see more and more campuses in India take this kind of shape. There are already some other examples of similar things. We were the ones to do it first, but I think it is now a model by which we can work. So this is sort of the description of the campus. And of course, needless to say, scientists within this campus are extremely friendly with each other and work together with each other and attend each other's seminars, ask each other difficult questions, go for beers together, whatever. I mean, they are very, very collaborative, and there's a certain joy in being part of such a campus. That they go to each other's canteens and things like that. Let's see. So what is research? I've told you where research is done, but what is research? I looked up the dictionary and I sort of like the definition they have. It's a systematic investigation into and study of materials and sources in order to establish facts and reach new conclusions. I've highlighted new because I'll come to this in a little bit. Now, there are many kinds of research. If you follow the history of science, etc., there's a kind of research where you go into an unidentified, sort of uncharted territory in whichever field, and you don't go in with a specific set of questions, but you go in with a specific set of observation tools. So, for instance, if someone like Diane Fossey went into Rwanda to study gorillas, and she didn't really have any specific questions in mind. Her main idea was to go in and observe what happens there and to then report it. And through those observations, she was able to piece together a great deal of how gorillas live. Jane Goodall did this with chimpanzees. And to a lesser or more degree, a lot of us will end up doing things like this, where we just sort of go in and explore an uncharted territory and see what happens and observe closely and report it. Then, of course, there is sort of the descriptive, phenomenological, or empirical. I mean, these terms are used interchangeably. Research, where what you do is once a discovery is made, then you can sort of take it further. You can systematically characterize it. So in the Diane Fossey example, it would be characterizing all the different behavioral interactions that she saw between the gorillas, and then tabulate them. Make sure that people understand the system better through those reports. And of course, this will be true in any field of research, in any topic that one can think of. So there'll be always this kind of work that is extremely important, and many times also the engine for new discoveries, because sometimes with resolution you stumble on really interesting discoveries. And then there is the third kind, where you actually go into the system. This is the explanatory, causal, or mechanistic research. We go into the system and you pose specific hypotheses based on what you know about the system, but you have some sort of a causal mechanism in mind. And then what you can do is you can go in and try and address whether or not those hypotheses are consistent with your observations. And this kind of system, of course, allows you to open the black box of a question and start to ask what's inside the black box, and how does it connect with each other. Sort of like looking at under the boot of a car and looking at all the machinery there and saying, okay, this is how it all connects with each other, so I know how it works. And this is true, of course, in any field, including, of course, in chemistry. So in all of these different kinds of research, there is plenty of scope for discoveries and reaching new conclusions. I'll give you one example. I'm going to try this out. But let me start by saying that I'm going to give you an example of some research that happened just down, a few doors down, my lab in Berkeley. I picked this one because it's a particularly nice one. This is of gecko adhesive mechanism. So a group of biologists or biomechanists at University of California, Berkeley, discovered or started looking into how geckos run up and down a tree any which way, and this was an old question that was posed by Aristotle a long time ago. And the question he asked was, how is this possible for a gecko to do? Why doesn't it fall? I think many of us have seen these wonderful creatures on our walls in Bombay. Certainly, I remember from my childhood, had a lot of geckos. We called them lizards, but they're really geckos. So what they did was they asked, what is interesting about the foot of this animal? And they looked closely at the feet, and they kept looking closer and closer. And what they found was that the feet of the gecko were extraordinarily lamellated, meaning they had many, the surface area of the foot was highly enhanced because it sort of came down to little hair-like structures. So then they asked, well, do these hair-like structures contribute to generating the forces, and how? And, after some extraordinary bit of interesting techniques that they invented, they showed that, in fact, they could build little force sensors, and they could show that individual setae could generate forces because of van der Waals forces, and they exerted a force, and if you multiplied this force by all the number of millions of setae that were in contact with the wall, then it was possible to generate the force. And a gecko can literally hang on the wall with just one foot or even just one toe. So this is really effective. This paper was published with much fanfare in Nature in the year 2000. And immediately following that, there were a lot of engineers who got interested in this. And what I'm going to show you now is a little video of one such effort. And I just want to make sure that the audio is, can you hear the audio? Can you hear the audio? Probably not.
No.
Yeah. So let me see how I can share the audio. And just tell me if the share sound. Okay. So I'm going to now play it again and tell me if you can hear the audio. Yeah.
In the quest to build a better climbing robot,
Can you hear the audio?
Yes, you can.
Okay. All right. Now I'll play this video.
In the quest to build a better climbing robot, mechanical engineers at Stanford chose an unusual muse, the gecko.
We talked to biologists and we said, well, okay, what animals are out there that have really excellent wall climbing abilities, and they're fast, they're agile, and so on. Gecko stands out. They can run at 1 meter per second, up, down, in any direction. They can work on both smooth and rough surfaces.
Grad student Elliot Hawks, along with other researchers, developed a substance that, like gecko feet, can easily attach and detach to surfaces repeatedly. And it works without magnets or electricity.
The adhesive is made out of silicone rubber, so it's not actually sticky. It has a cool property that when you just touch it to the glass, it doesn't feel tacky or sticky at all. But when you put your weight on it, it then engages.
The invention led to a superhero moment for Hawks. These pads, equipped with the adhesive and supports for his feet, turned Hawks' hands into gecko toes, letting him scale this 12-foot wall, albeit slowly.
That had been a goal for a couple of years, and it was very exciting to get up there and look down and be like, all right, we did it. If we lift it off with a rubber band doesn't really even stretch. But now we come down here, wrap the rubber band around the loading point, and pull.
The key to the adhesive is a sea of tiny wedges invisible to the human eye. This silicone model shows what a single wedge looks like enlarged 100 times.
When you bring it into a surface, initially they're like so, and because it's just the sharp tip of the wedge, the contact area is very small. So there's no adhesion. They're not sticky. But now if I apply a shear force, they all bend over like this. And suddenly the contact area is much larger.
This microscopic feature gives the adhesive its unique properties, but makes it challenging to produce in larger quantities.
We make this adhesive. We make the mold by hand, we do micro machining, and then we cast the polymer into the mold, and we make little patches, 4 by 4 inches or 10 by 10 centimeters at a time. It's sort of a handcraft.
NASA is exploring using the adhesive to grab space junk. And the CEO of a car company sees the potential to streamline windshield installations.
They use suction, which leaves a big circular suction mark at the end, which he then has to do a whole other step to clean afterwards. And since ours doesn't leave any residue, he was very interested in that.
But don't cling on to hopes for tapping your inner Spider-Man. Stanford isn't looking to license the technology for any climbing devices. In Stanford, California, I'm Sumi Das, CNET.com for CBS News.
Okay, so what I've shown you is a piece of research that went from idle curiosity about how geckos are able to climb up walls, something that we've seen all the time, to something that becomes technology that is super applicable. Now the question I have, and I'm going to maybe ask for a show of hands, is God, how do I see people? Yeah, a show of hands for whether you think number one is research, the first story, or number two is research. Say the other question is which is innovation and which is research. So is number one research? Just a show of hands. I don't know if this will work. This is better done in person, I think. All right, and no one seems to respond. So I guess I'll just go ahead and just say that most people will say that, look, the first story that you showed was research. And then the second one, where you saw some technical applications of it, is innovation. And my answer to that is actually no, that is not the case. In fact, both of these examples have tremendous amount of both research and innovation. So what I want to say is that research is innovation and innovation is research. And the reason I say that is because, I can tell you from first hand experience, I happen to be a few doors down this lab, and I have seen and contributed in discussions where we, with the point of discussion of how do you measure something. So you built little devices to be able to measure, and then you built little strain gauges that allowed you to be able to measure the forces that were fine enough, etc. I mean, there was a lot of interesting inventions that went into the first example, where where they came to the conclusion that geckos could climb walls because of these van der Waals forces. And likewise, in the second case, although the primary aim of the video was to tell you about the technology that was invented, I can tell you that there was a lot of research that went into making sure even simple little things, to put them together requires you to try out various different materials, and things have to be just right. And that requires a lot of research. It's not all about technology. A lot of it is about just fundamental research. So at my, after sort of being in this kind of field for a while, I don't see the real difference between research and innovation. And this is good news. So what do you need for research? I forgot to mention that, I'm happy to take questions at any time. Feel free to interrupt me if there are questions. This can be quite informal. Okay, so what's required for good research? Well, more than anything else, you need students, lots and lots of students who are enthusiastic. They have hunger for knowledge and food, joy of discovery, and scholarship. Students who are willing to go deep into the literature to look for what has been done so far, and where we need to go, etc. Of course, just having students doesn't help. You need resources. You need to provision them with space, materials, money, facilities, infrastructure, without which really there is no research. And again, this can only happen in an environment where there's encouragement of free thinking, openness of ideas, a culture of scholarship, all of which we try very hard at our campus to make sure that we encourage students to be open about the ideas. It's not about being too protective of your findings and sort of keeping it to yourself and not discussing that. That is the death of research. Then, of course, you need to have opportunity. I mean, opportunity is always important, especially in our country. We really have a serious problem with providing opportunity to all. So access to knowledge and research spaces, and we make the institute available to internships and things like that, just so there will be some ability to share the resources that we have with whoever would like to use them. And of course, you need stamina. I mean, research is not a marathon race. It is a marathon race. It is not a sprint. Unfortunately, our entire sort of exam-dominated structure makes you believe that it's a sprint, and that answers have to be there on the tip of your tongue. You have to be able to summon them right away, etc. Whereas research is all about finding out, at its own pace, whatever the pace may be, what the answer is. And so all of these are required, and I'm sure I've forgotten a few other requirements, but these are the main ones in any event. And so, this is what one is trying to develop in a campus such as ours, where we're trying to make sure that we do well at all of these different things, and make sure that there's a lot of openness and discussion. A common question that I get asked by students, it's typically phrased as such: is there scope in research? There's a common idea that research is sort of a sort of esoteric pursuit that people who are of a certain studious nature follow it. But I mean, if any of you were to visit the campus, you'd know that that's not the case at all. You get all sorts of really wonderful people doing research, many of them with very interesting hobbies and things like that. But is there scope in research? Well, it depends. It depends on what you want out of life. Certainly, a research life is not formulaic. It's not a 9-to-5 job. It requires constant learning. You have to be on your toes all the time. You have to never stop being a student. And that keeps you young, excited, and outward looking. You meet interesting people all the time. You stay immersed in science. You travel to places. And it's really a wonderful thing to be able to do that. It's also an interesting thing personally, because your friends are your collaborators, are also your critics, are also your mentors. It's all rolled into one. And so it's one of the few professions in which this happens. It also allows you to think in a multidisciplinary manner. Of course, we've, as humans, categorized topics into chemistry and physics and biology and so on, but nature doesn't really respect these boundaries. And we just have to go where the question takes us. And there's plenty of physics in chemistry, as you all know. There's plenty of chemistry in biology, as you all know. There's really no clear boundaries between these. Are there jobs? Well, I think there are lots of jobs. There's a lot of new institutions, ICERs and NICERs, and IITs are expanding. There's been a general growth in infrastructure, certainly in the in the last decade. In fact, in the 2 decades ago, and those colleges and places, also new private universities are coming up, which are really wonderful in what they are trying to do. And so I think there's a lot of jobs and hiring, and teaching colleges are getting upgrading their research credentials due to the pressures of the NAC accreditation, etc. So research is now important, and colleges need to have a very proper research credentials, if you will. Salaries and stipends, I often hear that scientists don't get paid enough. That used to be true. That's not true anymore. I think the new pay commissions have done very well, both for students and for scientists. And I personally feel I get paid much more than I really need. And I know that this is true about many other scientists. But there's of course not enough for greed. There's never enough for greed. But it's plenty, and it's actually not bad at all. So I would say that that can't be the reason to not go into research. Governmental support is erratic. It depends a lot on other priorities. There's a lot of things like now we have the COVID crisis, so a lot of money was spent there. That there's only so much in the system, and so there is ups and downs there in the periods of plenty, and then there are periods of paucity. But by and large, there is some appreciation for fundamental science in the governmental structure. I can't say the same about industrial support, which is presently not a whole lot, except in certain areas, but it is growing. And I think the culture of philanthropic funding for research is entering our bloodstream. And so I think there is a chance of seeing more and more industrial support for research, although there's almost no appreciation for fundamental science when it is also connected with the industry. But I guess there's good reasons for that. And what does training in research help you with? How does it help you? I mean, when I say training in research, I mean typically like a PhD. What does a PhD really teach you? And a true answer to that question may sound almost trivial, but is actually very, very deep. Training in research teaches you how to read. We all know how to read, since kindergarten, but to really know how to read, to sort of read something deeply and make the right connections, etc., really require a certain kind of training. It also teaches you how to write. So much of our time is spent writing and making sure that we say nothing incorrect and make the right conclusions and state them properly. How to think, logical thinking is the center of scientific research. And so it is very, very important for us to be able to think very clearly about anything we're doing. Then, of course, are the things that people typically think of research as, which is the methodology and the data analysis, etc. We have to communicate the research in conference presentations, publications. And then also, and I think this is really important, especially in modern times, is how to think about what's going on in the society. I think all of us read newspapers and Facebook and Twitter or whatever. And there are within these media a lot of conflicting information, what I call facts and alternative facts. And how do you tell the difference between what to put your trust in and what not to put your trust in is something that I think a a person that is properly trained in research has a better understanding of, simply because they have learned to think logically and in a data centric way. So, this is really what I had to say about research and innovation, which is, that I think a career in research is actually a really wonderful thing. The days when I can scarcely believe that I'm getting paid to do this, and I really think students should think about this very, very deeply. What I'll do now is actually talk a little bit about my research, which is not in chemistry, but it is something that I thought I'll just give you a glimpse of, because it has many of the elements of what I talked, is, we really ask curiosity-driven questions, and we just study them. And sometimes they've gone on to do well in the technology side of things. But the lab has a very DIY feel to it. We build everything ourselves, and we really enjoy doing that. So the two parts to my research, one is insect flight. So I'll just give you a brief glimpse of what we do in this particular part of my research. So what I'm going to show you is a movie that is from the lab, and I should tell you that this movie takes a really long time to arrive at. What you're going to see is something very simple and something that I'm sure you've seen many times in the canteen. You're going to see a common housefly as it lands on the ceiling, okay? Except what you're going to see here has been shot with a high-speed video camera, and it requires us to understand the insect to a degree where we know exactly where it is going to land and how to create the conditions that allow it to land. So I'll just play the movie, and I'll tell you why this movie is important for us in any case. So here's the fly. You can see that the legs are extended, so it knows it's going to land, and it does this really wonderful maneuver, and then lands on the ceiling. Now, why is this interesting? To us, a movie like this, contains all the joy of a phenomenon that we want to study. First of all, this fly flaps its wings 250 times a second. Okay, so that's something to store, about 4 milliseconds per wing flap. So you can almost never see the wings in motion. And the fly is doing many things and doing them extremely fast and extremely precisely. And so one of the questions that we've been asking in the lab is how do flies or other insects operate at such rapid timescales and yet in such a precise manner? You normally know that, for instance, if you're a bowler and try to bowl fast, your accuracy is failing. And if you try to be too accurate, then your speed is failing. But insects are able to do both. And of course, they're able to do both because they have a very interesting brain. So inside that body is an absolutely stunning brain about which we are getting to know more and more every day. And that brain is integrating information from the sensory modalities, meaning the fly is actually watching where it is landing. So it has eyes, compound eyes, as you might all remember from high school. And then it has and the eyes contain, bring in information. It also has a vestibular system, which allows it to balance in 3 dimensions. Now the brain takes information from both these sensory organs and integrates it, and then, makes very quick decisions about what it needs to do with its wings. So that's essentially what I'm going to show here. So if we want to study insect flight, then first we have to find out what is the context. In ecophysiology is what I call that context. So insects typically fly when they're looking for mates or food, or tracking odors, or navigating over long distances, or guarding territories. So that's sort of the first part, is where it involves the exploratory side of things. Okay, so you go out in the nature and you look for interesting things that are going on. And then once you pick one of them, now we get to the part where you have to describe this behavior. And so you take a behavior, let's say it's looking for an odor plume, then you can recreate that behavior in the lab and then study it. And then you ask, okay, what are the sensory organs involved? These feed into the central nervous system. They generate a motor pattern. So they call the muscles to twitch in specific ways. It's like a concert with the CNS as the conductor of the concert. And then the muscles will then pull on the various cuticular parts, which are the sort of the musculoskeletal system. And that causes the wings to move in very precise ways. And I can go on and on about this, but it's a really fascinating system. And when the wings move, this is where I got into this problem, was through my physics background, was when the wings move, it pushes the flow around, or the fluid around it, which in this case air around, generates the locomotive forces. And so this requires, of course, physics, knowledge of physics to be able to decode this. And many years ago, we did this using a robot that we constructed, and that allowed us to make many findings that I won't go into right now. So a simple action that you saw requires all of these things to work out. Okay, and we can't really draw boundaries around any of these questions and say, I'm only going to study this, because then you're not addressing the entire problem. And so then, once we discover a behavior, we bring it to the lab, we use high-speed cameras, we reconstruct it in 3 dimensions. Okay, so, we're doing different kinds of assays that allow us to observe this behavior very closely. So in this case, we're looking at just how a fly turns. So, here comes a fly, and it's going to pitch up, so it will go up. And we would like to know what it's doing with its wings to be able to generate this maneuver. And so we are able to digitize the wings, etc. I won't go into too much details. This is the same fly, not the same fly, the same kind of fly, doing a yaw turn. The one you saw before was pitch. So we can do different things. But all of these assays allow us to generate this, and then we can reconstruct this thing. And then, we write programs that allow us to simulate this. And this really is required for us to be able to understand what's going on with the control and dynamics of this insect. So, we then recreate it, model it, etc. And then we have specific findings. For instance, we can show that this is where a turn occurs, and we can look at the wing motion and say exactly which movements. I can't go into details of this in a short time, but this is what it requires. And so this is again another film. In this case, we have digitized everything and put all the plots down. And as you can see, the animal is approaching a vertical object this time to land on. It's going to land. These are 2 views, so we need 2 views to reconstruct it in 3 dimensions. Now, there's a part of lab that studies the mechanics of all this, the physics and mechanics of all this. There's also a part of the lab that records from single neurons in the brain, and then ask what are those neurons doing and how are they, taking in information and then sending signals to the motor system. And that system is very complex. So if you look at, this is a micro CT reconstruction of the insect. So, this is X-ray tomography. And so this is a fly. And I just wanted to show how complex this simple little animal is. So we're going to now go inside the fly. You're going to see all the different muscles lined up. So these are all the different muscles, and you can see all these flight muscles right here. There's also steering muscles right there, and then the base of the wing, which is a really fantastic structure. And then there is, some brain is right there and in the head, which you can see very clearly. And because of tools like this, we are able to make novel discoveries, because we're seeing the fly in a very different way than we used to in the past, when you had to sort of pull it apart, with dissection tools. And another question that we are really interested in how do you take all of this and pack it into a really tiny insect? And just to give you a sense of how tiny, this is one of the smallest insects in the world, called Megaphragma. It's a wasp, and its size is smaller than many single cells. This is a paramecium, and yet it is perfect. It is able to fly. It is able to sense with its antennae. Its wings are weird looking, but they do the job just fine. What I'm going to show is a video of an insect flying. And again, getting to this video is really difficult, requires all kinds of inventions that I can hardly describe. But they are able to fly just fine. And lastly, another topic in the lab is insect architecture. So we're looking at all these different structures that insects, especially social insects, build, and asking how do they build it? What's going on with the collective building? And so the specific thing we're working on is termite mounds. I'm sure you've seen this in and around Bombay. And these mounds are built by millions of little termites that look like this. They don't have eyes, and they don't have, they obviously don't have a blueprint of what they're supposed to build, yet they're able to build these really intricate structures. I can show you some images from the inside, and trying to understand how they're building this. How do they know what to build, how to go around this, navigate in absence of eyes? And of course, it shouldn't surprise you that chemistry is involved in this problem, because a lot of the communication that's going on is through chemicals that the termites secrete. And we are now trying to understand the chemistry of this. So this is just a video of the termites at work. You can see that they're building this mound block by block with little boluses of mud that they regurgitate. And there's a lot of really interesting physics also involved in how this mud binds together, etc. A lot of biology. The brain is very useful, and as I mentioned, the termite has to know exactly where to pull mud out of and where to deposit it. And these are two opposite behaviors, yet they know what to do and where. And that's of course being guided by chemistry, chemical signals, pheromones, etc. And so we bring this assay into the lab, and here's a time-lapse video of the termites at work. And so it's assays like this that allow us to recreate the behavior within the lab in much more controlled environments, and then make the discoveries that we need to make. So just a movie, and then I'll end, and then I'll take questions. So here's a movie.
Termites are what motivated this whole project. We learned about these termites, these centimeter-long insects that together build mounds that can be 40 feet or more tall. And we said, that's amazing. That shows that these large numbers of independent agents, all with limited sensing and no central control, that they can together build very large-scale, complicated structures. That's fantastic. Now, how would we do that? How would we build and program robots so that they build according to these same kinds of decentralized principles, but together produce particular things that we ask them for? This video is sped up 5 times because the robots actually move very slowly. They're capable of moving faster, but we have them moving very deliberately. They're being very careful to get everything right. Sort of measure twice, cut once. So here we've sped up 15 times. The robots are actually moving at about 2.5 feet a minute. So the main window is showing sort of a 3/4 view of the 3 robots building the structure. In the lower left is an inset showing a top view of the same thing. So if you're a human asking these robots to build you something, you start by giving them something like a picture of what you want. You give them basically a blueprint. And a computer takes that and annotates that with the equivalent of traffic rules. that gives the robots guidelines on how they're allowed to move through the workspace. Each one has its own brain, doesn't know what the other ones are doing. They're not getting directions from any central command. And they also each have all their own sensing. They can only tell what's going on right around themselves. They're working together in the sense that they're all working to build the same structure, but they're working independently. Each one is acting on its own. If you got rid of one of these robots, it wouldn't affect what the other two were doing. If you got rid of two of the robots, the third one would be able to keep going and finish the job by itself. The long-term vision of a system like this is to do automated construction in settings where humans can't easily do it. So for instance, if we want to send astronauts to Mars, it's very hard, very dangerous and expensive to send astronauts to build there. It would be ideal if we could send a team of robots on ahead to build them a habitat, so that would be waiting for them when they show up. Of course, that's, again, a very long-term vision. Something that might be shorter term, still on the order of years, but maybe not decades, is to have a system like this build levees out of sandbags to protect against flooding. Classically, the times when you want to use robots instead of people are situations described by the 3 Ds, dirty, dangerous, and dull. And something like building with sandbags in a flood zone is all of those things. Right now we send in the National Guard. So if we could have robots do that for us, that could help us keep people out of harm's way. Again, not these specific robots, not these exact algorithms, but you can imagine how systems like these could be adapted to build walls out of sandbags to protect against flooding.
Okay, so that was Justin Werfel, somebody that was part of a group that I worked with on this project. So I'll stop there and I'll take questions. Thank you.
Yes, sir. Thank you.
Yeah, thank you, Sanjay. It was refreshingly a different talk from what we chemists normally get to hear about. The floor is open for discussion. Students, you all can put up the questions that you have. Jisha, you can start.
Ma'am, with what? With the vote of thanks?
No, the questions.
Ma'am, we have a question on YouTube and I'll be sharing it in the chat box.
There's a question on?
YouTube.
Okay, okay. I think Professor Gunvanti will be looking at it, I think. Okay. There is a question in the chat box, Sanjay. Let me read it out. Okay, there is this. To begin with, what are your thoughts on the major energy source that will be utilized in the future? And how long do you think the current energy sources that we have will last?
It's a bit out of my expertise. I don't know. I think ultimately sun, I guess. That is the one that will last the longest.
Yeah. Solar energy will last.
Yeah.
Okay.
So there is a question on YouTube.
Mm-hmm.
As a researcher, how to set yourself apart from others?
Uh-huh. Yeah, that's a really good question. That's a difficult question. Part of the answer is that, in fact, nothing you do is really new. I mean, people have thought about things for the longest time. And so, typically one is, as Newton very nicely put it, standing on the shoulders of giants. So it's not, so I think one shouldn't try too hard to look for new things, but what really makes a difference is thinking about things with a fresh perspective. And often that's where new discoveries come from. When you're not thinking about the questions in the same way as has been taught before. What happens is, and this is exactly where I think multidisciplinarity helps. When a chemist thinks about biology, they're thinking about the question very differently than biologists would. Or if a physicist is thinking about biology, they're thinking about the question from a very different perspective. And this is one of the reasons why we feel strongly about, us crossing fields and trying to think of things in a different way. So if you want to really do things differently, you have to think about things differently. That sets you apart. Of course, new technology also helps. I hope I answered.
Okay, so there is one more question from the student.
Mhm.
If we want to make a career in research, then when exactly should we start working for it?
Now, anytime that the muse strikes. I think, it's a never-ending process, really, and it doesn't have a definite start. I mean, you can start by reading books and getting into the mindset of discovery and how discoveries are made, etc. You can start by just going to some lab or writing for internships, and then sort of requesting. My only advice there is to make sure that when you write to the labs, to not write a generic email. I mean, I often get emails from students that say, yeah, dear sir or madam, I want to work on cancer. Can I join your lab? And obviously my lab doesn't work on cancer. And, I would just ignore an email like that. So it's very important when you write to people to have done your homework about, what that person works on, whether this is interesting to you. If so, why? Articulate that in your email, and you're already, on the road to being a researcher.
Okay, thank you, sir. Similar question by one student on what would be the qualifications required for doing research?
Well, I mean, depends. So, if you want, I typically think of research as a research career as starting somewhere in the last year of your master's program. At least for me, that was the case. And that is really when I worked on a project, but nowadays you can start it even earlier. You can do summer internships, etc. And for that, the real only qualification you need is enthusiasm and persistence, and is a background in a similar topic. At the master's level, of course, you're doing something towards a curriculum, so you that's part of a program. Once you've done your master's, then you can apply to graduate school or for PhD, and for that you need an MSc. If you're in the US, then 4-year bachelor's degree also is allowed to start research. So that's sort of the qualification you need. Typically, for to be a postdoc, you need a PhD, and to be a faculty member, in most institutions, you'll need a PhD or at least an MSc is very important. Both of these, give you sufficient expertise to teach a subject, but I think typically also to do research.
There's one question in the chat box. Like a student is asking, what are the challenges a researcher will face during his career?
Right. Lots of challenges. For instance, many times okay, let me step back a bit. If you're doing it correctly, research is really looking at new things. Okay, so you should not be able to find the answer in any book. If you're doing things right, you're looking for something that is not in a book. You will be the one writing the book on that particular question. So when you're really looking at something new, as is true in every aspect of life, and this was something that Dr. Vaidya just mentioned in his really thoughtful lead up to this talk, is you'll make errors, you will make a lot of mistakes, you will learn only through failing. I think this is true about everything we do, but this is especially true about research. I think none of us would have learned to walk if we hadn't fallen a whole bunch of times, or learned to ride a bicycle without falling a bunch of times. I think the same is true about research. So, you try and you fail, and you try and you fail, and then you try and you succeed. And then, then you have a result, and then you take the next step. And this is the process. Now, what you require to go through this process is stamina, as I mentioned. And many people lose the battle by just sort of running out of steam. And the tougher ones, keep going and are able to actually reach the end. It's not as hard as I might make it sound, but that's really what it's about. Many times results come easily, that's not a problem. And many times they may take a little bit longer.
There's one question in the chat box. Are bees more intelligent and productive than humans?
I'd like to think so, but actually that question can't be answered, because one has to look at all these things in their proper context. I mean, there are things bees can do that humans can't do. For instance, they can fly, they can sense things really fast, they can make very rapid decisions, and they can, they are able to build these extraordinary hives, etc., etc. But then there are things humans can do that bees can't do, like solve crossword puzzles or whatever, or build bridges or things like that. So each thing has to be seen in its proper context. And I'll actually go as far as to say that every animal has to be seen in its proper context. And jellyfishes might not seem like much, but in their own environment, they are really, really good at what they do, which is why they've survived as long as we've survived in the evolutionary history. I mean, we are all of the same age, more or less, if you believe that life started with one cell.
Okay, so there's one question in the chat box about what are the tools which you have used in your field research.
Okay, let me see. Let me count all the tools that we've used in my field of research. In my lab, one of the key things that we use is high-speed videography. So we have these high-speed cameras that are able to film things in very, very slow motion. Then we use electrophysiology, which involves being able to record from single neurons. So you need really fine electrodes that are filled with a ionic solution that you can insert inside a single axon of a neuron and record from it. So that's something we use. And of course, connected to that entire system is a huge amount of electronics. And so you have to know a fair bit of that. You need confocal microscopy, because what we do is we fill the neurons with dyes, and then we have to image those dyes. These are fluorescent dyes. So you have to then dissect and take the prep to the confocal microscope, sometimes a 2-photon microscope. Then there is electron microscopy that we do. I showed you one movie of micro CT or X-ray microtomography. That's another tool. a lot of chemistry because we have to make solutions and so on. What else? There's a lot of tools we build. Like I can give you one example, which is that to be able to film at very high speeds, you have to throw a lot of light at the system because the amount of light coming through the lens for very small exposures is very little. So you have to throw a lot of light at the system, and in the process, you might just fry the insect that you want to actually film. And so we devised a system, for instance, which is able to turn on the light only when the shutter is open. And so it's sort of connected with the camera system. So it is only on for a fraction of a second, only when the shutter is open. So those kinds of things we have to do. I mean, there's just so many different things that we just build in the workshop to make the assays possible. But these are some of the key ones.
Okay, thank you, sir. A similar question, one student has requested to share one of your research that you find most fascinating.
Of the things that I have done?
Yes, sir, yes.
Oh, okay. I mean, God, everything is fascinating, and it's all connected. But right now, my mind is really occupied with the question of how neurons encode information. So, our eyes, for instance, see a continuous world, and yet, they don't encode all of that information. There's a lot of information they throw out. For instance, we're not encoding ultraviolet or infrared cues, but even, we're not encoding at the rates that we need to encode at. So there's a lot of compression of information that's going on. I mean, imagine that you're seeing a picture in 10 megapixels, but it turns out that you can shrink that picture to maybe 1,000 pixels without really understanding that there was any difference. And that means that the brain doesn't really need all the information that is out there, so it doesn't need to encode everything. And so one of the things I'm thinking a lot about is this sparse encoding of information as it enters the brain. So this is one thing. And then I'm also thinking on mechanics of flight a lot. I don't know, maybe that was too detailed an answer, but those are the kinds of things I'm really keeping me awake at night.
Okay. So there's another question which says, if you are on a research, how can you demonstrate that your solution is a good one or it satisfies the topic of your research?
Okay, that's a really good question. Let me interpret this. So, how can I demonstrate that I've got the right answer?
Yeah.
Yeah. So, that's what, I talked about, the hypothetical deductive process. And that's really what it's about.
Yeah, better.
So what you do is you propose a hypothesis and you then test the hypothesis. What you try to do is disprove the hypothesis. You can't really prove anything in science. What you can do is disprove hypotheses. And then if your evidence is consistent with the hypothesis, then you can actually, sorry, there's some an open mic.
Yeah, but this rain water, I was thinking about it. Single tap.
Yeah. Okay. Thank you. So what you do is you then test this hypothesis, and if your evidence is consistent with the hypothesis, then it stands, wins the day. But it may not. There can always be evidence that proves it wrong another day. But that's how science progresses.
Okay, thank you, sir.
One last question. There is a student asking, how can students get enrolled in international research programs?
Depends where you want to go. If you want to go to US, then you have to take the GREs, but GREs are not really required anymore in most places. But they have, I mean, each university has their own enrollment processes, and they are similar. And so you can apply to them. In Europe, also they have a screening process. The ads come out and you can apply to them. Different places have different processes. What else? Australia has some different way. It's been a while since I actually applied to graduate school. So, but I mean, these are the standard processes for each of these countries, and you can apply for them. What you do need to demonstrate these days is some background in research. So you should do internships. I would encourage you to do internships, and you should have a very clear idea of what you want to do, because very often when they interview you, they can really tell that you really, if you're confused or not clear, then they can tell, and that usually doesn't go well.
Yeah, thank you, Sanjay. There are many questions, but some are from the chemistry side
of things. You can answer them better than me, I think.
So that maybe I will answer them in the classroom tomorrow.
Mm-hmm.
Okay. Gunmanti, you can take over now.
Thank you, Dr. Sane, for addressing all the questions put in by our students. Curiosity, as we know, is the driving force for all the research and innovations, and all the questions put in by the students have shown that the session has shown some, has directly
I think we lost her.
Yeah, I think she has lost the network, I think.
Mm-hmm.
Thank you, Sanjay. I will just request Jisha Xavier of TYBSC to propose the vote of thanks.
Good evening to one and all present over here. It's an honor for giving me this opportunity to be proposing the vote of thanks for this event. I, Jissa Xavier of TYBSC, would like to express my sincere gratitude to our esteemed speaker, Professor Sanjay Sane, for taking out time from his busy schedule and enlightening us with thought-provoking knowledge on research and innovation. Thank you, sir, for introducing us to the fundamentals of research, how research and innovation goes hand in hand, and also some fascinating researches like the gecko adhesive mechanism and also the motility in the common houseflies. And I'm sure that you have definitely awakened some innovative minds. I would also like to extend my gratitude to our founder, sir, Professor A.E. Lakdawala, Management Principal, Dr. Vikas Vaidya, and the Department of Chemistry for organizing this informative event and for always being a guiding light to us. Last but not the least, I would also like to thank our audience for their active participation and making this event a successful one. Thank you all once again.
Thank you very much for inviting me. It has been a pleasure.
Thank you, Sanjay. Thank you.
And if there's anyone who wants to ask me questions, they're welcome to email questions to me.
Yeah. There are some questions, but then maybe we can have it at some later time. Maybe.
Sure. All right. Thank you.
Bye-bye. Thank you.
Thank you.
So we'll end the meeting. You all can leave, students.
Yeah, end the meeting.